Hey everyone, I'm going to get us started. Welcome to the fall seminar series. I'm Steve Seibold. I'm an affiliate with the department, and I work for the U.S. Forest Service here in town. And I have the pleasure of introducing Egan Chen, who works in our lab. Egan, of course, was trained in China. He got his bachelor's degree from South China Agricultural University in plant protection. He also got a master's there in agricultural entomology. And then he went to the University of Georgia, where he did a PhD on tritrophic interactions with beet armyworm, cotton, and a parasitoid. And the key question there was, how does nitrogen fertilization affect those interactions? And he did a little transitional thing. He was in agricultural entomology and started working on pecan trees, and I think that's how we hooked him on forest entomology and tree insects. So he did a brief postdoc there at Georgia, and then went on to Michigan State University, where he worked in our sister laboratory in the Forest Service in East Lansing. And there he worked on emerald ash borer, which is one of our primary invasive forest insects now. It's a major, major pest. We've dumped a lot of money into that. So he worked on nutritional ecology, a little chemical ecology with emerald ash borer. And kind of attracted our attention because we have a similar species here, the gold-spotted oak borer. So when his project ran out there, we were able to snag him, and he came here to Davis in 2011. And has worked for several years on gold-spotted oak borer, published several really good papers, and has moved our program forward. More recently now, he's working on the Polyphagous shotwell borer, which is another pest that's coming to Southern California, invasive. This one's from Asia, and it's an ambrosial. He'll include that in his presentation. So his talk today is going to web all that together. He's been here long enough that we've promoted him to an associate project scientist from postdoctoral scientist. And he's going to put all those pieces together in this presentation, interactions of invasive wood-boring beetles with their host hardwood trees. Egan? I'll turn the lights on.
Thank you, Stu, for your detailed introduction. Good afternoon, everyone. Thank you all for coming. And I would also thank the department and Dr. Stu Netherland for the invitation. It's a great opportunity for me to share some of my research in the past 6 years. And today I will cover 3 invasive wood-boring beetles and use them as examples to illustrate some specific insect-plant interactions. So the first beetle is emerald ash borer, EAB, and its host plants are ash trees in the genus Pheromones, and it's now in eastern and midwestern USA. It was introduced from northeastern Asia. And when I was working at Michigan State University as a postdoc, I was interested in nutritional, chemical, and behavioral interactions between EAB and ash trees. So today I'm going to show you one of the interesting behavior of EAB, larval feeding behavior, and its driving force. And the second beetle is gold-spotted oak borer, GSOB. It was the host plants are oak trees in the genus Focus. It's now in San Diego County, California. It was introduced from southeastern Arizona. And for any invasive species, first critical step for eradication is to detect them early. So in the past 3 years, working at UC Davis, we focused on visual and chemical interaction between oak trees and the GSOB in an attempt to develop trapping lures for early detection. And the third beetle is Polyphagous shotwell borer, and this beetle attacks many hardwoods, and it's an ambrosial beetle, which means they feed on a fungi and instead of plant tissues. It's now in Southern California. It was introduced from Southeastern Asia. So we started this project this year, and I will show you our preliminary results on host range testing and plant signal-based loop development. So what's common about these 3 beetles? First, they are all wood-boring insects feed attacks main stem. So according to this book chapter by Bill Mason, who is a prominent forest entomologist, and wood-boring insects that are attacking main stem are most damaging among all insect-feeding yields. EAB alone has killed tens of millions ash trees in USA. So I will show you two examples here. One is the EAB, and this compares the ash trees 3 years after the introduction of EAB, and all ash trees on that tree has been wiped out. And the second picture shows oak trees killed by post-live oak trees killed by GSOB in San Diego County. And what's common about these 3 videos, they all attack hardwood trees. As we know, hardwood trees are well known for their strength and resilience, and they are widely used for flooring, furniture, and so on many other uses. And many hardwood trees are also very important as street trees. For example, ash trees comprise 5 to 10% of street trees in North America. So let me first talk about the emerald ash borer EAB. And emerald ash borer is so called because the emerald color of the full body. Here is an egg. Eggs hatch, then tunnel through the bark and make a S-shaped galleries. And EAB has 4 larval stages, and they overwinter as a pupa. And the EAB is native to Asia, northeastern Asia, including China, Japan, and Korea. In North America, it was first discovered in Detroit area in Michigan, probably through woodpecking material. And later the same year, it was discovered in Ontario or Canada. And in USA, since its introduction, has been spreading to neighboring states. For example, in 2003, it spread to Ohio, 2004, Indiana, and to 2013, it spread to the westmost states in USA and Colorado State. And to the south, it spread to Arkansas and Georgia. Host plants, all ash trees native to eastern US, they are susceptible. And some species, examples of eastern species are white ash, black ash, green ash. These 3 are most widely distributed ash trees in USA. In western and southwestern USA, there are more ash species, but their distribution is much more limited. And examples of western species are single leaf ash, California ash, Oregon ash, chocolate ash. So the EAB attacks trees of all sizes, so diameter ranging from 2 centimeters to 1.5 meters. This slide shows the distributions of ash trees in natural forests. So I want to stress its distribution in natural forests. The distribution in urban settings is not available. So as we can see, the most widely distributed trees are 3 eastern species. Black ash is mainly in northeastern USA, and white ash in eastern half, and green ash is distribution is wider. In the western species, we have single leaf ash mainly distributed in Utah, and there are quite few in California. Velvet ash mainly distributed in Arizona, and also some in California. So in California, we mainly have California ash and Oregon ash in West Coast, Northern California, and Sierra Nevada. So this seen from this picture, California is connected to the westmost states that EAB has been detected, Colorado, from the ash species. So it will be interesting to see if in the next few years EAB keeps spreading to the west, either through natural disposal or human-aided introduction, as shown in this cartoon. And usually, human-aided introduction was much faster than natural disposal. Here are the EAB damage symptoms. The D-shaped egg circles is related to the characteristic, and many euphorestids, like the good spotted oak that Aviatopicus second, also has the same shape as the pole. And woodpecker damages, woodpeckers are after EAB larvae, clown dieback, all these branches have been there. Sometimes one will see many epicomic shoots below inside. And the internal symptom or S-shaped or zigzagging damages. Here is an illustration, cross-section of water structure. The parts that the EAB feed include vesicular cambium, living fluid, and pork cambium. And the consequence of damage to these parts is that the nutrient and water transport is interrupted. And as a result, trees usually will kill in 3 to 4 years. And in a study looking at how the EAB larvae affect ash tree chemistry, I found that most of the larvae hatched from pinned eggs, they move downward to the root area. So here, the panel A shows the y-axis, the number of larvae, and y-axis of the two moving directions, and significantly more larvae move downward to the root area. Also, the biomass of the larvae moving downward is much greater than those moving upward. And in field collected trees from 2 parks in Michigan, and we observe the same behavior. So I want to know what's causing the downward moving behavior. And the answers to these questions might help us understand what selection pressures that EAB larvae may face during their evolutionary arms race with ash tree. So the observed behavior is not because of gravity, because when seedlings were grown upside down, as we can see here, most of the videos moves upward, which is the root area. So these 2 spots, dark spots, indicate place that the eggs were pinned, and they are moving upward. Here shows that more larvae move upward. And the observed behavior is not because of polyphenols. Polyphenols is a really bigger group of plant secondary defensive compounds that they can bind to soluble proteins and make them insoluble and invaluable to insects. So this picture axis is the duration of EAB larvae feeding from 10 days up to 50 days of continuous feeding. And y-axis is the ratio of total polyphenols below the feeding site to above the feeding site. So samples were taken right below the feeding site and above the feeding site, then compared. So a ratio less than 1 as shown by undamaged control trees like the black bar, the white blank bars indicate the concentrations of total polyphenols is less below the feeding site is less than above the feeding site. However, EAB damage after 40 days continuous feeding, the ratio was increased to about 1, which means the total phenolic concentrations is greater below the feeding site than above the feeding site. And another line of evidence, polyphenols not being a causing factors that EAB larvae can excrete the polyphenols to fluoresce to the majority. And this figure shows x-axis is tree species we tested. Y-axis is the total polyphenol concentration, either from phloem, which is the black bars, and fluoresce, shaded bars. So majority of the polyphenols goes to fluoresce. And also regardless of the larval stages we tested. And moisture content might be one of the causing factors. As we can see, for undamaged control plants, the ratio is below the feeding site to above the feeding site is close to 1 consistently. EAB damages increase the ratio to greater than 1, so which means the moisture content below the feeding site is greater than above the feeding site. So I want to see if moisture content affect larval development. And I set up an experiment with 4 different moisture content using artificial diet. So I have 20, 40, 60, and 80% of moisture content. And the panel A shows the mortality of larvae that lived on artificial diets with lowest moisture content, 20% is greatest. And the panel B shows, in contrast, the biomass of the larvae lived on low moisture content, 20 and 40% are the lowest. So obviously, moisture content is one causing factor. Nutrients might also be one of the causing factors, and 9 essential amino acids and 3 non-essential amino acids. The ratio was changed by EAB larval feeding. So essential amino acids are amino acids that we humans or animals cannot manufacture on our own. We need to get them either from plants or other source. And this shows one of the non-essential amino acids, prothionine, and the ratio significantly has increased. Also, the artificial diet study indicates that nutrients plays important role for their development. So I have 4 treatments, and one is control. The control has both casein. Casein is a protein source, and the yeast is a amino acid source. So control has both of them, and neither. The treatment has neither of them. So no casein, the treatment has yeast, and no yeast has protein. So they have either one of them. And panel A shows the mortality again is highest on the treatments without casein nor yeast. In contrast, the biomass is the lowest for the diet without casein and yeast. So to summarize EAB section, since its discovery in 2002, EAB has been spreading really fast, and it now has invaded half of the USA. EAB has killed tens of millions of ash trees and may keep spreading to the west and to the south. And EAB larvae move downwards during their feeding and due to moisture content and nutrient availability. So let me switch to good spotted oak borer GSOB. And good spotted oak borer has very distinctive 6 golden spots on the elytral. As EAB and GSOB has 4 larval stages, and they overwinter as late stage larvae near the bark surface. And GSOB was first discovered in San Diego County in 2004. However, it was not associated with oak mortality until 2008. And genetic analysis indicate the populations in San Diego is from southeastern Arizona, likely through firewood. Distributions in California, and it was spreading slowly in San Diego County, but it's still to 2014, it's limited to San Diego County. Although in November of 2012, there are a few trees found infested with GSOB, but the trees was removed shortly. And after that, there are quite intensive surveys, and they found nothing. So far, it's still restricted to San Diego County. And damage symptoms very close to EAB and D-shaped example, wood damage, and clown dieback, as straight galleries. And one is different from EAB is bigger staining you will see on all trees. And oak trees are broadly divided into red oaks, white oaks, and intermediate oaks. In oak woodland in southern California, in the field, one will find red oaks, including coast live oak and California black oak, are much heavily infested than intermediate oak, which is California live oak. And there was almost no infestation on white oak incremental. However, in field, in laboratory using no choice test, larval feeding, EAB larvae, they feed on red oak, white oak, and intermediate oaks. Although the damages on red oaks are generally greater than intermediate oak, which is further greater than white oaks. So this picture shows the x-axis are the oak species, and the gray bars represent red oaks, black bars represent intermediate oaks, and white bars represent white oaks. The y-axis of panel A and C is a proportion of larvae that successfully established generally in no-choice condition. And the y-axis of panel B and D are proportion successful larvae that survived to the final stages. So the proportion is pretty high in all the panel, and can go about up to 25%. So at Dennis and using that campus, you will often see copper oak, which is an intermediate oak species, and valley oak, which is a white oak species. And in laboratory, GSOB, they feed on both species. So if GSOB keeps spreading to the north, these species might be endangered. And oak distributions in California. So this is a zoom in of San Diego County. In San Diego County, you will see all 4 oak species. So the yellow patch represents Ingleman oak. The white and the red one behind here is the California black oak. The blue one is Canyon live oak, and the green one the coast live oak. So as we move up to north, we will see more susceptible oak species like coast live oak, Canyon live oak, and California black oak. It's widely distributed in West Coast, Northern California, and Sierra Nevada. And GSOB prefers big trees. As we can see from this picture, none of the 8 trees diameter less than 15 centimeters was infested. As diameter goes up, the proportion of trees infested goes up too. And all of the 6 trees with diameter greater than 105 centimeters was infested. And for any invasive species, as I say, the first critical step is to detect early. So we focused on visual and the chemical interactions on this video. As we know, many videos use visual and chemical signals from plants for host location. And this picture also shows GSOB has a well-developed eye and antennal. So semiochemicals are chemicals that mediate interaction between 2 organisms. And the organism could be from the same species or different species. So for developing color traps, we have used 2 techniques. One is using an electrophysiological technique called electroretinographic analysis to measure GSOB sensitivity to light wavelength. Secondly, we scan the oak foliage of different oak species using spectrophotometer, then determine their optimal wavelength and match the color of to the wavelength. And this work is done with our collaborator of Damon Crook at the University of Massachusetts. And here is an illustration of the electroretinal detection. Beams or lights with different wavelength will cast on the head of an insect, and the electrical signal then was detected and recorded. Another electrophysiological technique that we used later on for screening chemicals is called GC-EAD, and this work is also done with our collaborative demo group. And GC-EAD is coupled with this chromatography, electro-antennal graphic detection. It's used for rapid screening of compounds in a complex mixture. And here is an illustration of GC-EAD, and samples were injected here, and then samples go through a very long column. They are separated here, then split into two halves. One go to a GC detector, so that we know what compound they are. The other half go to an antennal or an insect. Then the electrical signal was detected and recorded. The retinogram shows that GSOB are sensitive to quite a few of peaks. There are some in red area, blue, purple, and UV. Here are the colors that we developed. These names of the colors are from paint strips. So the result indicates that purple trap is the best color. So later on in our study, we used purple trap as a control. And then we also scanned the foliages from different oak species, including preferred host coast live oak, intermediate host Kenyu live oak, and non-host Engelmann oak. So here are the colors that we developed. This slide shows the results. As you can see very clearly, purple trap, regardless of male, female, or the total, is the best trap. Then we also look at semiochemicals of different sorts. First, we look at EAB Pheromone and the green leaf volatiles. I have two definitions here. One is pheromone are semiochemicals that mediate interaction between two organisms from the same species. And the green leaf volatiles belong to pheromones. Pheromones are semiochemicals that mediate interactions between two organisms from different species. So the pheromones benefit the receiver, but not the emitter. We also look at the pheromones from oak bark and try to isolate GSOB pheromone. So Peter Silk, who is a research scientist from Canadian Forest Service, and his group, they isolated a EAB sex pheromone 3Z lectin from EAB female, and also they tested its isomer 3E lectin, and both of them elicited an antennal response to EAB males. So because EAB and GSOB are in the same genus, they are pretty closely related. They want to test these compounds to see if there are any cross attraction. And in their study, they also tested two green leaf volatiles. One is 3Z hexenol and 2E hexenol. Both of them elicited an antennal response. And both of these GLVS are also produced by oak trees, so we want to test these four compounds. Their study indicated that 3Z lectin increased the male EAB catches, and both lectin isomers increased the male EAB catches to 3Z hexenol. So here is our study site at Cleveland National Forest, about 30 miles east of San Diego County. Here is a specific study site, and they are release rate. For the EAB sex pheromone and its isomer, we have 2.4 micrograms per day release rate. For the two GLVs, the release rate is 17 milligrams per day. And we also had 2x2 combinations of these four compounds. And they use the trap as a control. So here shows the results, and it's overall catches were very low. It's less than 1 video per trap for 2 weeks. And unfortunately, there's no treatment effect as well. And we conducted quite a few of the contrasts. For example, for this one, we compare the unbaited control with all the rest of traps that has either 1 or 2 lures, and it's not as significant. So after the field season, we got a chance to look at GSOB antennal response to EAB pheromones and to green leaf volatiles in GC-EAD. Results indicate that neither GSOB female nor males are responsible to 3Z-lectin, 3E-lectin, and 3D-hexenol. GSOB females are not responsible to 2E-hexenol. However, there is something interesting. GSOB males are responsible to 2E-hexenol. This compound is really shown here. So this result is both surprising and exciting. It's surprising because in the last season we didn't see any attractiveness of 2E-hexenol. It's exciting because it elicited antennal response in GSOB males, so we still got a chance to develop a good lure. And so we want to test this compound for 1 more season in the same national forest. And we had 2 different release rates, 17 milligrams per day as usual, and we increased the release rate to 102. And we also included ethanol, because many bark beetle and woodworm insects are attracted to ethanol. Ethanol is a sign of tree declining or fermentation. So here are the results. Again, the water catches are very low, less than 1 meter. And unfortunately, again, there's no treatment effect. None of the contrast we conducted was significant. So then we looked at the pheromones from oak bark. We collected the bark and then extracted the volatiles and run GC-EAD. Here are the GC-EAD response to the males. These are a list of the compounds listed in antennal response. So we tested some of the combination. One is bark blend, which includes 8 out of 10 compounds, and the camping along, and the camping plus gamma-tropinin. And as you can see from this picture, none of these loop combinations are better than unbaited control. So far we also collected volatiles from GSOB females and males, and preliminary results indicate there's some difference in the chemical profile here. So we are still now in the process of identifying these compounds. And if we do find a GSOB pheromone, that will be very significant. So to summarize GSOB section, since its discovery in California, GSOB has been spreading pretty slowly. And purple chapter is the best color we have now. Many volatiles from oak barks and oak foliages listed in antennal response, but they didn't do well in attraction of beetle in field. And now we are testing different combinations of different compounds, because in reality, insects they can sense a blend of chemicals. So we are testing 3 of the compounds with different combinations. Lastly, let me talk about the polyphagous short-bow bottle. Polyphagous short-bow bottle was first discovered in Los Angeles County in 2003. It's probably introduced from Southeastern Asia. And this beetle is very closely associated with a fungus called the Fusarium dieback. And polyphagous short-bow bottle females inoculated this fungus when establishing a gallery, and later on both adults and larvae feed on its fungus, so they don't have to interact with tree chemistries very intensively. Here are some pictures of the Polyphagous ball. This is a female. It's much larger, about twice the size of female male, and the color is also darker, and the males are flightless. And here are some different stages of the beetle. There's one egg here, different larval stages, pupa, and adults with male and females all in the same gallery. Distributions in California, it was first discovered in Los Angeles County in 2003. Now has spread to Orange County, Riverside, San Bernardino, and San Diego County. San Diego County was first detected in 2003. However, I have one picture I want to show you. This picture was taken this summer, and the sycamore tree has been completed. All the small brown spots are the extra holes. The point I want to make is that although it was detected in 2003, but the tree has been killed. That means the beetle has been in San Diego for a few more years before the detection.
Detected in 2013.
2013, yeah, I'm sorry. So if we did a survey earlier or more intensively, the distribution of GSOB might be greater, broader. So the symptoms take a different form on different host plants. And these ones on box elder, the dark color is caused by the fungus. And this on coastline oak, you will see a bigger stain. And on avocado, the white stuff is called they call the honey volcano. And on box elder, on castor bean, you wouldn't see stains, but on sycamore you will see lots of stains. So according to scientists from UC Riverside, they surveyed that over 200 species are known host plants. Here are some examples, and the species highlighted in red are the species you might see very often at Davis. And now we are still testing different host trees from different states, and based on the ability of Polyphagous to establish a gallery, and all these 6 species are susceptible. So in the study, we forced a female Polyphagous model to feed on a log. Then if they establish a new gallery, so in the gallery, in the beginning there's only one female. Later on you will see a male, female on different larval stages. Then we said they are susceptible. And the species followed by a red star sign indicates the species has not been reported in host. So we have found 4 more species in California, and the species from Louisiana and New Mexico, or these 7 species are susceptible. And Polyphagous for infestation in the field, infestation could go as high as 90% on box order and a below species, and the mortality could go up to 30% box order and over 20%, almost 20% on red willows and castor bean. So as GSOB, we want to develop effective rules for detection, and because box elder is a preferred host, so we first tested the box elder volatiles, and we had 4 treatments using the funnel trap control without any ethanol, the ethanol lure. For the 2 box elder treatments, we hollowed the log and filled either water and ethanol. Here is a picture of the study site, and the preliminary results indicate there's no treatment effects, although the treatment box aerosol plus water will attract more beetles numerically, and also the overall catch was pretty low. So to summarize prolific short-hole borer section, prolific short-hole borer is spreading pretty fast in California, and it could be a really biggest threat to hardwood trees due to its broad host range. And its complete host range is still unknown. We are testing different host trees from various states, and many species from Louisiana and New Mexico are susceptible. Prolific short-hole borer is not attracted to ethanol, a signal of plant fermentation. In the future, we are focused on collecting volatiles from different host trees and then test their efficacy. So to summarize my talk, many hardwood trees are endangered by many invasive food borers. And for the two Volatiles species, EAB seems to be spreading much faster than GSOB. One reason may be that EAB infests trees of all sizes, while GSOB prefers bigger trees. And also, the living phloem of oak trees is much thicker than ash trees. So the oak trees can sustain more populations of GSOB, and before they can move, they must move to new areas. And developing plant-based trapping techniques for waterboarding is very challenging, and so far purple trapper is the best color we tested, and no effective lures for prolific short-hole borer and GSOB. And both EAB and GSOB, and they limited their host range to trees in one genus, where, however, prolific short-hole borer has really broad host range, and perhaps because prolific short-hole borer, they feed on fungi, so they don't have to interact chemically with oak trees intensively as the other two species. So thank you for your attention, and I will take any questions you have.
Before we start the questioning, I want to say I neglected my introductory comments to note that Mary Lou Flint played a really big role in bringing him to us into European and supervision. That was a really major part of our team on G7. So I just forgot to say that. I'm sorry. So any questions for Egan?
Yes. What's your questions on the biology of these species? Do you know whether any of these produce sound?
No, what?
Do they produce sound? Do they make any sound?
I'm pretty sure the larvae, when feeding, they will produce sound.
But the adults do not. They don't have any wing vibrations, or any movement of the body that produces sound.
Is that for mating or
Possibly.
as far as I know, I'm not aware.
It's one of the means by which insects attract each other, including beetles. So, because the male will often be attracted to a female sound or vice versa.
Yeah, and I'm not aware. I haven't seen any report about the adults, but we did actually for the larvae, we recorded the vibration of different stages of the larvae, and the later the stage and the greater the amplification. So that's all I know about.
So any indication that the adults are attracted to larval sound?
The adults?
Are the adults attracted to larval sound? Which would be one way to increase the attack rate on a tree.
I don't know if they are attracted by sound or some other signals. It's possible by the decline of the tree. It's possible. I know GSOB, they prefer previously infested trees. So that obviously they have some signals actually.
And do the males multiply mate, or is it a single mating person?
Males may multiple times with a female. Females I don't know. I know males can work many times.
And so do they move in fact from tree to tree or from female to female?
For EAB, I know they move tree to tree, but for GSOB, we haven't seen any GSOB in the field besides beetles we caught in the traps.
Most of the adults are up in the crown of the trees up. Most of the adults are up in the crown, that's where the males and females.
Males take place in the
I
crown.
think so, and the females come down. Move positive.
Okay, the males stay up in the crown and multiply mate?
We think so. I mean, I don't know what the multiple meaning, that's probably been observed in the lab.
Yeah,
Well, the implication here is that, if males can mate with many females, then, the strategy would seem to be, in terms of trapping, would be, get the female. So you're aiming your trapping techniques at catching the female.
Yeah.
There's
a really strong visual component to this with EAB where they've worked on this a lot more. They've actually used models and pinned insects, and the males come down and sort of parachute down on the females so they see them. And that's why this purple color is probably so important for them. It's related to the color coming up in the females.
So when the females emerge, do they then fly from tree to tree, or do they just fly once?
I don't think anybody knows so far. We haven't seen any behavior. It's possible. I wouldn't say anything. I mean, nobody knows. I haven't seen any reports about this. But I know, I mean, as Steve said, that they use this visual for male finding. When you watch a video, female, the males will still land on the dead males. So obviously they are using visual signals for mate finding.
The males on the dead females.
Dead females.
Yeah. Any other questions? Getting back to sound, that ambrosia beetle, we don't know anything about that. A lot of our beetles stridulate. That could be that going on. But in this case, the males stay in that tunnel most of the time and mate with their sisters. And so the male doesn't disperse at all, doesn't come out on the surface. And so all the mating that goes on, the females really just need to bore in and start laying eggs. And they have been mated previously by their brothers. They don't really do much anything. Gallery. Their property and the mate was made.
So when they depart, do they depart after mating?
The females don't depart. They mate. Yeah, they depart after mating. Yeah. This is the brood departs.
But the idea of the larval, some vibrations or something actually attracting adult beetles to trees is, I think, a really interesting idea, which I don't think has been tested. Pretty sure hasn't been tested for GSOB, because we do know that they infest the trees.
Yeah, that's a good point. We've been sort of fixated on whether there's odors on those.
Yeah, no, I think that's a very interesting. But we never see the adults in the field. We never see them. I mean, it's amazing. We spend hours and days out there monitoring, and the only time we ever find them is in traps. So it's hard to work with these insects, the adults, because they're stuck there.
Well, there's also some notion that the vibrations of the larvae are attracting the woodpeckers. So the woodpeckers are foraging up and down these trees, and they're probably sensing the larvae that are from their sound. So there's sort of an heterotrophic level of interaction with sound. Okay, if there's no other questions, we'll thank Egan for a really nice. Oh, there's
I have one small question. So you said that PSHB is not attracted to ethanol, like a sign of fermentation of a dying tree.
EAB?
PSHB, Polyphagous shottle borer. I'm curious if there's a trend in the kind of trees that Polyphagous shottle borer attacks. Is there no, do they attack dying trees versus living trees? Is there no variation in that? Would that be a potential explanation for why they there aren't attractive to the ethanol, or they're attracted to the ethanol?
Seems like they will take healthy trees
Okay.
too.
Okay.
At least for like box elder, as far as I know. And I don't know if they prefer previously infested tree. But I know, I believe they will take healthy trees.
Okay.
So within the sort of spectrum of these ambrosia beetles, a lot of them like to climbing trees, fermenting, and they all respond to the ethanol. This one is more like a primary insect, almost like our pine bark beetles in Sierra Nevada. It comes into healthy trees.
Okay.
So it's pretty unusual ambrosia beetle, and that does, explain why they're not going after ethanol in our experiments.
Okay, great. Thank you.
There are a whole second wave of ambrosia beetles that do come to those trees, even though the shovel board is there, and they are going to the fermentation process later.
Okay.
So sort of later.
All right, that makes more sense.
That's a really good question. I was at a workshop yesterday, we had that same discussion.
Okay, great.
Okay, well, thank you.